Hello students, welcome to Rushmi's classes. Class 11 Biology. Chapter 12 Mineral Nutrition. Mineral nutrition. Definition of mineral nutrition in plants. Carbohydrates, proteins, lipids, water, and minerals are required for all living creatures to survive. Plants, too, require nutrients for growth and development. Methods to study the mineral requirement of plants. Hydroponics is a method of growing plants in nutrient solutions rather than soil. This method is employed in the commercial cultivation of vegetables. It's also used to research plant mineral deficiency disorders. Aeroponics is a method of growing plants by spraying nutrients floating in the air. Essential mineral elements. Mineral needs vary depending on the plant. There is a criterion for determining whether or not an ingredient is necessary. It contains the following items. The element must be required for proper development and reproduction. If such a component is missing, the plants won't be able to finish their life cycle. The element's need must be precise, and no other element should be able to substitute it. The element must play a direct role in plant metabolism. There are two types of mineral elements required by plants: macronutrients and micronutrients. Macronutrients. The plants require a high amount of these components. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium are all found in it. CO2 and H2O are the most common sources of carbon, hydrogen, and oxygen. Others are received as mineral nourishment from the soil. Micronutrients. These are the nutrients that plants require in tiny quantities. Iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel are all present. Essential elements. Essential elements like carbon, hydrogen, oxygen, and nitrogen serve as components of numerous biomolecules such as amino acids, lipids, and therefore structural elements of cells, and are classified into distinct groups depending on their unique activities. Essential elements, such as magnesium ion in chlorophyll and phosphorus in ADP, are key components of chemical compounds associated with energy. Essential elements operate as both activators and inhibitors of enzymes, influencing their function. Both ribulose bisphosphate carboxylase oxygenase and phosphenol pyruvate carboxylase are activated by Mg2+. Both of these are required for carbon fixation to occur. During nitrogen metabolism, Zn2+ activates alcohol dehydrogenase, while MO is required for nitrogenase activity. The osmolarity of a cell is controlled by a few key components. The stomata open and close in response to potassium ions. Role of macronutrients and micronutrients. The role of different macro and micronutrients are: nitrogen. Plants require a considerable amount of this essential ingredient. It is taken by plants as nitrate ions, NO3, with certain plants also absorbing it as nitrite ions, NO2, or ammonium ions, NH4+. It is an essential component of vitamins and is required by plants for actively developing tissues such as meristematic tissues. Plays a direct role in photosynthesis. Phosphorus. Phosphate ions, PO4, 3+, are absorbed by plants. It is found in large quantities in cell membranes, proteins, nucleic acids, and nucleotides. Encourages the development and growth of roots. Seed formation needs the presence of this nutrient. Involved in the storage and transport of energy. Potassium. It is rich in actively developing tissues such as meristematic tissues, buds, leaves, and root tips. It is necessary for sustaining osmotic potential in a cell that is responsible for stomatal opening and closing. Increases photosynthetic rates. The protein synthesis is dependent on it. Calcium. Calcium ions, Ca2+, are absorbed by plants and are required for ongoing cell division and development. Plays a part in nitrogen metabolism. It exists in the form of calcium pectate in the middle lamella. It is also necessary for the mitotic spindle to develop. It is also necessary for the activation of certain enzymes. Magnesium. It is necessary for the activation of enzymes involved in respiration and photosynthesis. It is absorbed by plants in the form of magnesium ions, Mg2+. It aids in the production of RNA and DNA. It's a crucial component of chlorophyll, as well as maintaining ribosome structure. Sulfur. An essential component of amino acids like methionine. An essential component of amino acids like methionine. Contributes to seed production. It is necessary for chlorophyll creation. Iron. Iron is absorbed mostly in the form of ferric ions, Fe3+, by plants and is necessary for cell division and development. Assists in the transport of electrons during a variety of metabolic processes. It is necessary for the activation of the catalase enzyme, which is essential for chlorophyll synthesis. It also functions as an oxygen transporter. Manganese. Manganese is absorbed in the form of ions, Mn2+. One of the components of the nitrogenase enzyme, which is essential for nitrogen metabolism. It is engaged in the photolysis of water during non-cyclic photophosphorylation. Zinc. Zinc ions, Zn2+, are absorbed by the body. Essential for the production of chlorophyll. Plays a role in the activation of numerous enzymes, including carboxylases. Required for carbohydrate synthesis. Copper. It is absorbed in the form of cupric ions, Cu2+, by plants. During metabolic processes, it is engaged in redox reactions. Performs photosynthesis and reproductive functions in plants. Contributes to the flavor of fruits and vegetables. Increase the amount of sugar in the plants. Boron. Boron is absorbed in the form of Bo33 or B4072 by the plants. It plays a role in calcium absorption. Assists in the development of pollen tubes. Plant carbohydrate transport. Plays a part in cell differentiation. Molybdenum. It is obtained by plants as molybdate ions, Mu2, 2-. Nitrogenase enzyme's main component. It, together with iron, forms the enzyme's catalytic site. It's a crucial part of the nitrate reductase enzyme. Nitrate reductase is a nitrogen assimilation enzyme. Nitrate reductase is a nitrogen assimilation enzyme that also plays a role in nodule development. Chlorine. Ions of chloride are absorbed, Cl1. Maintains the cell's osmotic potential. It is engaged in the photolysis of water, which results in the production of oxygen. Deficiency symptoms of essential elements. One of the most crucial necessary elements is nitrogen. Yellowing of the leaves is caused by a nitrogen deficiency. Phosphorus shortage results in burned leaf tips and yellowing of the tips. Potassium shortage results in interveinal chlorosis and wilting of older leaves. Blossom end rot is caused by a calcium shortage. Magnesium causes older leaves to become yellow, a condition known as chlorosis, which occurs when chlorophyll is degraded. Sulfur shortage causes younger leaves to yellow more than older ones. Chlorosis of young leaves is caused by an iron deficit. Dieback disease is a common symptom of an iron shortage in plants. Manganese causes yellowing between the veins of the leaves and creates dead patches on plant components. Zinc causes the plant to develop rosettes, yellowing between the veins, and reduced growth. Copper shortage results in the weakening of cell walls, as well as stem and twig dieback. Boron has an effect on plant reproductive and vegetative development, as well as meristem death. Molybdenum causes growth retardation. Chlorine produces wilting, chlorosis, and a highly branching root system, as well as necrosis in the tissues. Metabolism of nitrogen. Nitrogen is made up of two nitrogen atoms that are linked together by a triple covalent bond, NN. Nitrogen fixation is the process of converting nitrogen into ammonia. The conversion of nitrogen to ammonia can be done in two ways. The following are the details. Physical nitrogen fixing happens when lightning strikes. In the presence of lightning, N2 and O2 in the atmosphere react to create nitric oxide, NO. NO is then oxidized further to create nitrogen peroxide, NO2. Ammonification is the process of producing ammonia from the decomposition of dead plants and animals. Nitrification is the process of converting ammonia to nitrite and ultimately to nitrate. Nitrite is formed in the presence of Nitrosomonas bacteria, whereas nitrate is formed in the presence of Nitrobacter bacteria. The microorganisms do not take place. Found in rainy season during lightning to know the microorganisms do not take place found in rainy season during lightning to know. Lightning. N2O, nitric oxide, 2N2O2. 2NO2 oxidation. Nitrogen peroxide. 2NO2HO2. HNO2 4NO22HO2O2 4HNO3. Nitric acid. CaO2HNO3 equals CaNO3 2H2O. Calcium nitrate. HNO3NH3 equals NHNO4 ammonium nitrate. HNO2NH3 equals NHNO4 ammonium nitrite. Biological nitrogen fixation happens when nitrogen-fixing microorganisms like rhizobium are present. Diazotrophs are prokaryotes that fix nitrogen in a biological way. Rhizobium is a symbiotic bacteria that dwells in the roots of leguminous plants. Nitrogenase is the enzyme responsible for biological nitrogen fixation. Both free-living and symbiotic nitrogen-fixing bacteria exist. Azotobacter, Rhodospirillum, Anabina, and Nostoc are examples of free-living nitrogen-fixing aerobic bacteria. Leguminous plants, such as peas, beans, clover, alfalfa, and others, have a symbiotic relationship with rhizobium. Nitrogen fixing is accompanied by the development of distinctive nodules. The contact of rhizobium with the roots of the plants is the first step in the development of nodules. Certain compounds are released by these plants, which attract bacteria to the plant's roots. Rhizobium produces a root hair curling component that aids the bacteria's invasion of the roots. The nitrogenase enzyme, which catalyses nitrogen fixation, is oxygen sensitive. During nitrogen fixation, the enzyme like hemoglobin, a member of the hemoglobin family, scavenges oxygen. Nodule formation steps. Bing images. Steps of nodule formation during biological nitrogen fixation. The reaction catalyzed by the nitrogenase is as follows. Nitrogenase N212 ATP ADP 2NH312 ADP 12 Nitrogen ammonia phosphate, atmospheric, inorganic. It requires 12 ATPs to break down the strong triple bond that exists between the two nitrogen atoms of N2 gas, NN. The process of nitrogen fixation requires a lot of energy. To transform one molecule of nitrogen into two molecules of ammonia, 16 ADP molecules are required. Plants are poisoned by the ammonia produced during nitrogen fixation. As a result, this ammonia undergoes further reactions. Reductive amination is the reaction of ammonia with alpha-ketoglutarate acid to produce glutamic acid or glutamate, as indicated in the diagram below. Reductive amination and transamination. Transamination. When an amino group from one amino acid is transferred to a keto acid. Transaminases are enzymes that catalyze this process. Asparagine amino acids are formed as a result of this process. As a result, poisonous ammonia is transformed into necessary amino acids for plant growth and development. Thanks for watching. Please like, share and subscribe.